Welcome, everybody.
There we go.
This is our second webinar for our cloud products. And today we'll be talking about using BigQuery to explore and search SRA metadata that we've made available. Before we can get started with the actual presentation, I would like you to take a poll for us about your familiarity with SQL and BigQuery. You can just click on your screen to answer the question, and once we get some responses, I'll close it and share this with everybody. And this kind of helps me to tailor the webinar to your needs so that I can go into more detail about certain things or in less detail if it's not needed. So it looks like most of you have heard of SQL, but haven't actually used it. So we have another poll for if you've used BigQuery. Besides just helping tailor this to the webinar, it also helps us to understand your use cases and if the products we're building for you meet your needs. Okay, so it looks like most of you have heard and used BigQuery, and for some of you, this is the first experience you'll be having with it. So I'm glad to be able to explain it. So quick introduction for our agenda. So I'll go through SRA in the cloud and what BigQuery is. And after that short introduction, I'll jump straight into the actual demo of the three use cases we'll be presenting today. So SRA in the cloud, what is it? At this point, we've moved all of our data to the cloud, and we have deposited more than 12 petabytes of data into the cloud. And this number grows daily. But having all that data, it doesn't actually help anybody, since you need a good way of actually searching for the right data sets that you want to compute on. And this is where BigQuery comes in. This is our first cloud native product for doing search for data sets. And it has two major benefits. One is its fast response to the queries you will run, and the multiple methods you can use to access it. It has a web interface, it has a command line API, it has multiple libraries that you can use in your programming languages like Python. And also, getting started with SQL is fairly easy. There's only a handful of concepts you need to understand before you can dive in and start exploring it. So if you are interested to learn more about SQL, Google has made a lot of efforts to provide documentation for BigQuery. They have query syntax explanations, they have tutorials, they have how-to documents. Besides what Google provides, there are online tutorials and web classes you can sign up for. Some of them are free, some of them are paid, where you can learn SQL as well. You can also search forums for questions on how to write queries. One of the popular ones is Stack Overflow, where you can submit a question and people will help you answer it. But there's also lots of questions being asked already, so you can certainly reuse those answers that's provided. So BigQuery is just a giant data warehouse, and we've deposited two data sets into it. The first is the SRA metadata, which includes sample information, how the sample is sequenced, and some basic statistics on the sequencing run that was performed. The second data set that we have is the SRA taxonomic content analysis. This analysis is done using a K-mer approach, and we predict species that are within a particular run based on these unique K-mers. And you can take both the results of both of these data sets, combine them in a query, and this will help you have a more filtered result depending on your use case. So a quick overview about SQL. So there's a select statement that tells that we're going to be extracting data from tables. There's the from clause that will tell the database from what tables we're going to be extracting data from. There's the where clause that specifies the filters we're applying to those tables. And a common one to use is order by, which helps to order the data set based on a particular column. Something maybe like release date or accession numbers. There's other there are operators used in SQL. Some of these are like the equal sign. Others that you'll see in my queries are the in operator, which you can use to provide a list to a particular column. There's the unnest operator that helps you flatten an array into its own table. And there's the like operator, which allows you to do pattern searches. So you'll see those in my use cases. So we have 3 use cases that we're going to demo. One is finding the taxonomic distribution of human gut metagenomes. The second one is finding coronavirus data within SRA. And the third one is identifying runs for Arabidopsis gene expression studies. This one, I'll just jump straight into the actual demo. So here we have my first query, which is the finding taxonomic content of human gut metagenome. So I'm going to just start running it now because it takes about 30 seconds to run. But to go over the syntax here, so I'm selecting only 3 columns. I'm selecting the accession column, the name column, and a column called self count. The self count column represents the number of k-mer hits. Now, in the from clause, I have 2 tables. One is the SRA metadata, where I'm providing the alias for it, M, and I have the SRA tools tax analysis table, and the alias for that is tax. So the aliases just make it easier to write queries where you don't need to rewrite the entire table name every single time. I'm then joining the 2 tables based on the accession columns. I'm providing a list of organisms to search in the metadata table for the organism column. And I'm looking for only public data sets. These data sets are publicly available and do not require any permissions to access. BigQuery does have dbGaP data, and so in order to make sure I don't need to apply for special access for dbGaP studies, I'm just looking for public data. And then I'm filtering the taxonomic results to provide me only the results for the species. The taxonomic analysis table contains the entire taxonomic tree for a particular species that was identified. And if you're interested in seeing the whole tree, you can certainly build that out. But for this example, I just want the actual species living in the human gut metagenome. So a couple things you can pay attention to on the web interface is they give you identify how much data will be processed for the query very easily here on the right-hand side. And they provide a quick check that there are no syntax errors in your query. So this is kind of a quick check you can do by eye to make sure that your query is fine. So now that we have our results, we have our accessions column, the name for the species that are within this accession, and the number of k-mer hits. So looking at this, we can also see that this result came back with roughly 40 million rows. So this is a lot of data here. And you can quickly scan through it by eye if you want, but there's really no way to scan through 40 million rows. So of course, the next step might be, how do you visualize something like this? What do you do once you have this? How to visualize the k-mer hits in an easy to understand manner? And there's a couple ways you can do this. The first step, though, would be to actually save these results to somewhere. Unfortunately, Google does provide certain limits to what you can save to a file directly from running a query. So in this case, there's a 1 gigabyte limit, or if you're saving to a local file, it only will save 16,000 rows. However, there is an option to save the result as its own BigQuery table, and this is what you will actually need to do. Once you save the result as a BigQuery table, then you have a couple different options which you can do. You can then send that entire table to Google's Data Studio and use that to visualize the data, build some graphs and dashboards for the data. Or if you want to run your own statistical analysis on the data, you can export the entire table as a file. Now, this is why you need to save the result as a table so that you can send an export command either through the web interface, command line, or through one of those libraries I mentioned for programming language to export the entire table into a file, into a Google bucket. Now, once the export is finished, you'll have a file that you can work on. However, if you do want to get that file to your local computer, you will need to pay egress charges to download that data from your Google bucket into your local computer. So depending on your use case and where you want to perform your analysis, those are some of the different options available for you to work with such a large data set as this. However, this can provide you some input about what species live in human gut metagenomes. So I think this is very useful. So now we'll move forward to our second use case. And you can see that I saved all of my use cases as queries, and this is very easy to do. You can just click Save Query and Save As, and then just provide any name you want to it. So for our second use case, which is finding coronavirus data set, I can just open it in query and run it very quickly. So this is a much smaller data set that we're querying, and so it will come back significantly faster. So in this case, what I'm looking for is the bio project, the biosample accessions, the run accession, the collection date, the geolocation of where the sample was collected, who it was collected by, and the host of the sample. And just like last time, I'm querying both the metadata table and the taxonomy analysis table. And what I'm searching for in this case is the Coronaviridae family. So instead of looking for a particular species or all species, I'm providing it a particular family that I'm looking for. And as you can see, we have lots of good metadata, like in these couple of first examples, where we have a collection date, the location, who it was collected by, and the host. But as you can see, there are some data sets that don't have a lot of metadata. And this is one of the challenges with working with user-submitted data, is that data you would expect to be there might not be there because it was never actually submitted to NCBI, or in this case to EBI, since this is a DNA accession. However, you can use these data sets to identify runs that are going to be useful for your downstream analysis. So in case if you are interested in human-based hosts, you can certainly then pull out the SRA accessions, save those to a file, and feed those accessions to the SRA toolkit to dump FASTQ files or SAM files, and then run your computational tools on those FASTQ and SAM files. To make things easier within BigQuery, BigQuery provides a lot of kind of useful tools and shortcuts. One of these is they provide an autocomplete feature. So to just quickly demo that, it would be if I started writing something. In this case, I want to maybe filter the data set based on when the data sets were released. So I'm gonna search the metadata table, and then I just hit tab, and then I can see all the columns within the metadata table. And then if I just start typing what I want, which is the release date, it will autocomplete and select the date for me. So in this case, I can quickly find the column that I want using this autocomplete feature. And if you want to learn more about shortcuts, BigQuery does have this little shortcut tool that you can look through and see the different shortcuts available for you. So in this case, if I wanted to filter by release date, I can say I want all the samples that were released within this year. And so it makes it very easy to write your queries using this autocomplete feature. So you don't have to memorize all the columns available in all of our tables. So moving towards our last example here, which is finding or the data set for RNA-seq analysis. So in this case, this query is a little bit more complicated because I'm working with a lot more metadata submitted by users. So in this case, what I'm doing is I'm opening the attributes column within the metadata table, and this attributes column is an array. So I use the onNest operator to make it a flat table. And then I'm looking for all the keys with the name of tissue, organism part, or organism sample. And I'm providing that an alias called tissue. So this way I can quickly identify what the tissue was for the data set. Next, I'm looking for what the treatment was for this data set. I basically copied and pasted the above query and changed the keys to treatment, environment, stress, or condition for this sample. Now, you might ask, how do I know what keys to use in this array column? Well, that was the more complicated step where I looked through different examples of different samples, and I picked these out and I saved them as a list to be used in this query. So there will be some work trying to identify the correct keys to use because different submitters like to provide different attribute names when they're submitting metadata, so it makes it hard to standardize searches like this. So besides that, I'm also going to pull out all of the metadata columns, but I'm providing it the except operator here because I don't want these 5 columns to be displayed. So instead of typing out metadata, accession, assay type, and all the other columns I want to display, it's quicker to provide a list of columns I don't want to display. For the from clause, I'm providing it the metadata table with an alias. And what else I'm doing is I'm going to unnest, again, the attributes columns as its own table. BigQuery will join these 2 tables on the back end by itself, so you don't have to worry about that. And the filters I'm applying here is the actual organism name. And I'm going to search through the sample attributes looking for the keyword either leaf, root, or leaves. And then the other approach I took is metadata has this column called JATR, and this is the JSON attributes column where we took all the biosample attributes and provided them as JSON. So here I'm searching based on a pattern for the word drought. Now the percent sign implies that it will apply 1 or more characters before or after the word drought. So I'm doing a very broad search here. And to make sure that I'm pulling out the gene expression runs, I provide the library source as transcriptomic. You can also take a look at the assay type here is also set to RNA-seq. That could potentially be another filter you apply. So looking at the results, we have roughly 300 data sets where the tissue was a type of leaf or root with a treatment relating to drought. So having looked at this and maybe pulled out some other attributes to filter the data set, maybe by a sequencing platform or what library selection strategy was used, I can narrow the data set even further. At the end of the day, though, the idea is you pull out the run accessions and get the actual data to run some bioinformatics RNA-seq computation on it. So those are the three use cases I wanted to provide to go through the queries I'm using and potential use of the results. Now, running BigQuery is not free, but it is very, very cheap. So here I provide just a quick predicted cost for running these queries. As you can see, our biggest one was the human gut metagenome, where we extracted 40 million rows, but that only cost us roughly 40 cents. The coronavirus data set was significantly smaller, just 30 cents. And the Aradopsis example was only 10 cents. So getting started with BigQuery is very cheap as well. So we are always looking for feedback. The only way we know how to improve our tools and what we're doing is if the community speaks to us. So if you have suggestions for improvements, please don't hesitate to contact us. Just write to suggest at NCBI and provide us any feedback that you wish about either things we're doing well or things that we can improve to fit your use cases. The more we hear from you, the better we can provide the tools that you need for your research. And here are some general URLs that you can look for more information. So we have more documentation available about BigQuery and accessing data in the cloud at SRA. If you want to learn more about how to export large data sets, Google does provide directions for that. If you want to play around with Data Studio, there's documentation available. And please sign up for our YouTube channel as we hold more webinars and provide more instructional videos. All of those videos will be available there. So it's a good resource to have for you. And that's about it. Thank you.
Thanks, Yuri. So we have a few questions in the questions pod. There's been some questions about costs, and you did a good job of explaining what those particular queries cost, but there is a free tier, is there not, for Google? Could you run any of these queries on the free tier?
Yes, Google does provide kind of a free tier approach where the number of queries you can run is limited, but you can certainly do that. And if you sign up for BigQuery yourself, Google also provides you 1 terabyte of data to process free every single month. So it's possible to sign up and set a limit for yourself how much data you want to process and just stay within that 1 terabyte free limit, which I think might be a little bit higher than the free tier, but that part I'm not quite sure about. But there certainly is a free tier for BigQuery.
Okay, another question, actually there's a couple of related questions, and it has to do with the columns and the sort of keys that you search on. I think this is about the Arabidopsis example. So one question is, what happens if there is multiple key matches? What results do you get back? So for example, the information about treatment could be in 3 possible keys, I think.
Right. It is certainly possible. In this case, there wasn't any overlap between these 3 keys.
But it's possible that there could be.
Yeah, potentially that could be. So I would actually need to explore that exactly how this particular query would act that way.
And I think it was just a general question too, as well as how it would behave if that was the case.
Yes, this is the complication with having user submitted metadata where the keys can change and different submitters will use different keys to mean the same thing. So you need to be very careful about how you use those keys, and you might potentially run into an error regarding the result depending on how the query is structured. So it does take some playing around with the query to get the exact result that you want.
Okay, thanks, Yuri. Related to that is this concept of the user submitted data having some lack of control, I guess, is maybe one way of putting it. And someone wanted to know if NCBI had done any work to sort of group related columns so that it would be easier to people to make precise queries.
Right. So NCBI does try to standardize the metadata that we request from people during submission time. That certainly helps. We have not had the chance to do more work on standardizing that metadata, but it certainly is something that we're aware of that needs to happen, and we'll be working on it in the future to make this process easier when writing queries. But at this time, we haven't dug too deep into it simply because of the complexity of the task. But it's certainly something on our roadmap to do.
Okay, thanks, Yuri. Another question is about, is there a way to run a query? Suppose you run a query today, and then you want to run a query, say, a month later, but you want to only query the new data, the stuff that's come in the past month, to save some money, basically. Is there a way to do that?
Not yet, as far as I understand with how BigQuery works. It searches the entire data set for your results. You can certainly set limits, but set filters for when the data might have been submitted or released. And so your return would be smaller,
yeah,
but BigQuery will still search the entire data set.
Oh, I see. So that would be applied after the search.
Yeah.
Okay, another question, which has to be with sort of diagramming the database, I think, is there a UML or an ER model of the SRA tables?
So we provide the schemas in our documentation available at SRA. So if we go to the SRA landing page, we have SRA in the cloud here, and within the search BigQuery pages, we have links to the actual tables and some explanations of what columns can be used. We did not put together a full UML diagram, but you can also link our entire project in BigQuery web interface. If you go to Pin project here, and you would need to type in NIH-SRA-data store and click Pin. And then you would be able to take a look at what is available also within BigQuery. So here you can take a look at the tables and see what columns and types are available for all of these data tables from within BigQuery. But that's a good idea to build the UML diagrams.
Okay, thanks, Yuri. There's another question that I think you may have addressed a little bit in the last webinar. Somebody's asking if we have a pipeline available that they can submit data with, and it looks like they're trying to filter out contamination errors from the data.
I don't quite understand the question here. A pipeline to submit data, the metadata?
Yeah, I'm not sure. I think they're asking about contamination in the data they want to try to filter out. I'm not sure what kind of contamination, maybe human sequences and metagenome data or something like that.
Okay, so we do have a process for filtering out human data from data sets. And when the data set is submitted to NCBI, if the submitter contacts SRA and tells us they want to perform human filtering on their data set, we can activate that process. We have not built that into our submission interfaces yet to allow the submitter to do that themselves, but we can certainly do that, and we have a process for that. We are working on a publication for that process at this moment, and once the publication is made, we'll definitely be linking it to it in our web pages.
Okay, great, Yuri. I think that's very helpful. I think we've addressed all the questions, really. Thanks everybody for coming. Thanks very much, Yuri. And I will.
Thank you everybody for joining us.
I will go ahead and end the webinar.